So we're going to talk about Chapter 5, the physical examination of urine. So urine color, which is normally various shades of yellow, can range from anything from colorless to amber, orange, even green, blue, brown, or black. These color variations indicate the presence of a disease, process, or some kind of metabolic abnormality, or some certain type of ingested food or drug. These color variations can result from excessive physical activity or stress as well. So really it is important to know any change in color of urine that is the initial or only reason why a person will seek the medical attention. So when we talk about the characteristic color of urine, it is normally due to a pigment called urochrome, which gives it its yellow color. Now this is a byproduct of metabolism because urochrome is a lipid-soluble pigment that is present in the plasma and it's also excreted in the urine. So although urine colors can tell us a little bit about what is happening with the patient, let's just talk about some of the colors of the abnormal and normal types of urine colors. So an abnormal urine is one that reflects any kind of pathological process. Sometimes it doesn't have an abnormal color, but normally colored urine may contain certain types of pathological elements. So just because yellow is a normal color doesn't mean there isn't anything going on with the patient. So if you have a patient with a normal urine, but when you do the chemical process, you see large amounts of glucose, that can definitely indicate some kind of pathological process. But in contrast, if you have a red urine, this is often an indicator of blood and can result from the ingestion of beets. Okay, so nevertheless, urine color is a valuable preliminary assessment of a specimen. So many substances really are capable of modifying this color. The same substance can impart a different color depending on the amount present, the urine pH, and the structural form of the substance, which can change over time. Red blood cells is a perfect example here. So in fresh acidic urine, red blood cells can be present despite the yellow colored urine, or it may quickly appear red or pink. The color of urine depends on the quantity of red cells present too. And as the blood cells disintegrate, hemoglobin is released and oxidizes, which causes a yellow or brown type or even black pigment. Now let's talk about bilirubin. It's another substance that can contribute to the urine color as it's a byproduct of the breakdown of hemoglobin. It usually has a characteristic yellow color, but when it's present in sufficient amounts, the bilirubin imparts an amber kind of coloration. Okay? As well as upon standing, bilirubin can oxidize to biliverdin, which causes the urine to take on a greenish color. Now, some substances are colorless and normally do not contribute to the urine color changes, but upon standing, it can convert several colored compounds. So urobilinogen is a normal product of urine, but if it's oxidized, it could have an orange-brown pigment. A number of foods cause color changes in urine with no clinical significance. Now these are very highly pigmented foods such as beets, breath fresheners containing chlorophyll, any kind of candy dye, vitamins A and B can definitely produce a sort of orange type of color, and beets definitely a red type of color. Now please refer to table 5.1 for the urine color terms and the common causes that can cause these urines to turn a different color, as they will be on your next exam. So if a normal urine is shaken, sufficiently, you can see that there's a white foam that is forced to develop on its surface, but it does dissipate quickly. So the characteristics when we talk about urine foam is its color, its ease of formation, and the amount produced. And any of this can tell us if there are any kinds of proteins present. So moderate to large amounts of proteins, such as albumin in the urine, can cause a stable white foam to be produced when a urine is poured or agitated, which is similar to egg albumin. So the foam that develops is thick and long-lasting. Another type of foam is bilirubin. So when bilirubin is present in sufficient amounts, the foam will be characteristically yellow. Now again, these are specific and very distinctive evidence for those two types of products that are present. So those were the physical characteristics of the urine color. And now let's talk about clarity, which describes the overall visual appearance of the urine. It's usually assessed at the same time we do the urine color and refers to the transparency of the specimen. So often called turbidity, clarity describes the cloudiness of the urine caused by suspended particulate matter that scatters light. So there is a table 5 or 5.3, which talks about the clarity terms, the definition, and the possible causes. Please study this table for your exam. Whereas clear talks about all solutes present are soluble, and this is normally normal types of clarity we want to see with the urine. Now hazy or slightly cloudy, you see visible particles present, but you can read through newsprint when viewed through the urine tube. Now clarity varies here with the substances and the amount present, so you could see a small number of contaminants or white or red blood cells. Now when you have a cloudy specimen as seen in the third image there, you have significant particulate matter, and usually the newsprint behind it is difficult to read and view. Here you might have some crystals, you might have some epithelial cells, some fats, or any kind of microbes such as bacteria or yeast, or again some contaminants. Now when you have a turbid specimen, you cannot see at all through the specimen at all. You might have some mucus or pus, sperm, any kind of contaminants there. And again, you might have a lot of microbes that are causing that specimen to be cloudy or turbid. Now, historically, the urine color or odor led to the discovery of certain types of metabolic diseases. But urine odors, unless it's very strong or distinctive, are not really documented as part of routine analysis because it contains so many organic and inorganic substances or byproducts. So normally, urine has an aromatic odor that is typically faint or really unremarkable. However, if normal urine is allowed to stand and age at room temperature, it can become very odorous because of the ammonia due to the bacteria converting it from urea. Early ingested foods can cause certain urine to have a noticeable color, such as asparagus, or certain types of IV medication where you have a distinct type of aroma. Types of metabolic diseases may produce an aromatic, sweet or fruity smelling urine. So in some of these conditions, most commonly diabetes, in which glucose is present and cannot be used by the body fat, is metabolized and compensated. So here you can have a little bit of a sweet smelling urine. So when we talk about the concentration of a urine, we're referring to the amount of solutes that are present in the volume of water that is excreted. So normally urine is 95% water and about 5 to 6% solutes. So now this definitely varies based on a patient's diet, physical activity, and health. So a dilute urine has very few particles present per the amount of water, whereas a concentrated urine has more particles due to the amount or less volume of water present. Now, in the clinical lab, the concentration of urine is expressed as either specific gravity or osmolality. So when we talk about specific gravity, this is an expression of urine concentration in terms of density. So the mass of solutes present per the volume of solution. And it's usually a ratio of urine density to the density of equal volume of pure water under certain kinds of conditions. So the reference range for specific gravity is 1.005 to 1.030. So the greater the density of the urine, the larger the specific gravity or value because it's more concentrated. So we're going to talk about the ways that specific gravity is measured in today's lab by either the refractometer or the reagent. So when we're talking about refractometry, this is an indirect measure of specific gravity, and it's based on the refractive index of light, where the light passes from air into a solution at an angle, and then the direction of the beam of light is refracted and its speed is decreased. So here, the ratio of light reflection in two differing media is called the refractive index. And here is an example of a refractometer where you place one drop of urine onto the lens here, and then you use your field of view to look through the monocular and determine the diagram that's going to give you an interface between the two media. You'll get a line, and then you'll get your specific gravity as a value. Now, what are the advantages here? You need a very, very small amount. You only need 1 to 2 drops, and you have some kind of correction to temperature compensation. So, but in the viewing field, it's very distinct. So you can see between the light and dark areas before making the reading, and it's very easy to use. Now you can also use the reagent strip method, and this is the most common. Again, it's an indirect type of method that changes color, and it estimates the urine density based on the amount of ionic or charged solutes that are present. So anything such as sodium, potassium, chloride, and ammonia will produce charges that produce a color change. Now, when the reagent strip is wet, okay, the pad itself has a polyelectrolyte and a pH indicator at an alkaline pH. So when it's immersed, all of these protons will be released, and that will be proportional to the ion concentration. So what this means is released protons change the pH of the pad to result in a color change of the pad, which you can read. Now, osmolality is the concentration of a solute expressed in terms of osmoles of solute particles per kilogram of water. So an osmole is defined as the amount of a substance that dissolves to produce 1 mole of particles in a solution. A reason for doing the osmolality measurements are to evaluate the renal concentrating ability of the kidneys, to monitor any kind of renal disease, to monitor fluid and electrolyte balance, and also to diagnose the causes of frequent types of urination. So osmolality is really determined by measuring a colligative property of the sample. So these properties depend only on the number of solute particles that are present. The size and the ionic charge have no effect. So only the number of particles present as ions or undissolved dissociated molecules affects the result. Okay, so let's talk about terminology for urine volume. So although the amount of urine is excreted per day is a physical characteristic, urine samples are not routinely assessed for that. But in normal situations, Urine can vary from 600 to 1800 milliliters per day. Okay, so with less than 400 excreted at night. So when an individual excretes more than 500 at night, the condition is called nocturia, which is a feature associated with certain types of progressive renal failure. So, in consequence, with these, the kidneys lose their ability to concentrate urine. So what are you going to see here? The specific gravity of the urine is unchanged, and at the time, it will remain at 1.010. So this is the inability of the kidneys to change the specific gravity of plasma. It's called isothenuria, okay? And this is definitely a problem that's caused with chronic renal failure. Now, if a person excretes more than 3 liters of urine a day, that terminology is called polyuria. Okay, so this is due to an excessive water intake, hormonal imbalance, renal dysfunction, or some kind of drug ingestion. Now, when we talk about the term oliguria, this is a decrease in the urine excretion production of less than 400 mils a day. It can be caused by simple water deprivation, excessive sweating, diarrhea, vomiting, or any kind of condition that decreases the blood supply to the kidneys, and eventually leading to anuria if it's not corrected. So this is a complete lack of urination and very dangerous because if it's not relieved, it can lead to a fatality because of the accumulation of toxic metabolic products.